Yes,
the
yes.
number of peptides that is available. You are duplicating?
Yes,
or
we are
not?
duplicating.
Or not necessarily?
In this case, we are duplicating, but in that case, we can choose
Oh,
the
thank you
peptide
so
like
much.
10 more, it's already been covered, the timer there. So the timer will already be covering the 9 more.
You feel more yourself?
No, because it
Yeah.
shouldn't be.
Now you know who you are.
Yeah.
The timer could be better than the timer, so making, double the number of prediction is not interesting, so.
I think this can be specified according to problem. In some cases, people wanted to have overlapping peptide, and then they predict for 9 and 10 as well. And if you want to go for the less number of peptide, you can choose like 9 as default.
But if you choose two different cells, it's double the number of.
Double number of peptide are being predicted.
Okay, good morning everyone. I hope you can hear me at the back. Okay, so I will continue where Sandeep left off. We will discuss MHC Class II peptide binding prediction in this session. I think we are running over time, so I may go a little bit quick, but you can stop me. And I hope we can, leave the elaborate, detailed questions towards the end or to the individual sessions because we need to cover a lot of stuff. So quick questions we can take along with the presentation, and then we can move the detailed questions towards the end. All right, so here's a brief outline. I'll start with a brief introduction, and we'll go in detail on the Class II binding prediction tool, the web version. We have different versions. So we'll go in detail on the web version. Then along the way, just like Sandeep did, I will give the different IEDB recommendations and guidelines on selecting the top binders. And I will also go through how you can predict promiscuous binders, why we should be doing that. And there is a time for a short exercise. It's good if you can do that. I mean, just to get a hands-on experience, and you can ask us questions if you get stuck with something. And then there's a new, it's not new, it's been there for 2 years now. So interface for the Class I and Class II binding predictions called Epitope. So we'll take a look at that also. And then the other versions like API or RESTful interface and standalone. Okay. So Sandeep was talking about the Class I or endogenous antigen processing pathway. So here in Class II binding predictions, we are dealing with exogenous antigen processing pathway, or the antigens or allergens that are generated outside the cell and that gets inside. So for example, like antigens enter inside through inhalation, ingestion, or injection, like bacteria or allergens or parasites, etc. So the antigens enters inside the body, inside the cell, and it binds with MHC Class II molecule here, and then it presents the peptide to the at the surface of the cell, which then are recognized by the T cell receptors from CD4 plus T cells. So the basic structure and principles of the Class II binding prediction tool are essentially same as Class I, which Sandeep went through in details. So I may not be going in very detail, which are essentially same as Class I, but there are some differences which I will go in detail. So the differences in Class II binding prediction tools are essentially derived from the structural differences between Class I and Class II tools, molecules, sorry, MHC Class I and Class II molecules. So what are the main differences between Class I and Class II? So Class I are molecules, MHC molecules are essentially present in all nucleated cells, whereas these Class II molecules are present in professional antigen presenting cells such as B cells, macrophages, dendritic cells, etc. And in case of Class I, only the alpha chain participates in binding of the peptides, but on the other hand, there are two MHC encoded polymorphic chains in case of Class II, which are alpha and beta, and both these chains participate in the binding. And in case of alpha only, I said like only one chain impacts binding. And the binding groove is closed in case of Class I, and that's why the Class I normally can accommodate only shorter peptides. But on the other hand, Class II, the binding groove is open, so it can accommodate longer peptides, and it can extend beyond the groove, and the flanking residues, the residues at the end can also interact with the MHC molecules. And in case of Class I, the peptides are presented to CD8 plus T cells, and here in Class II, they are presented to CD4 plus T cells. And briefly about the nomenclature. So in case of class 1, you see only one letter, like the name is mentioned like this. So this represents the alpha chain. Whether it's A, HLA-A, or B or C, it's the alpha chain. So we don't normally say what's the B chain because it's constant beta 2 globally. So, but on the other hand, in case of class 2, except the DR, for DP and DQ, we mention both A and B chains. That's why the nomenclature is like HLA-DPA and DPB, or DQA and DQB. We are mentioning both alpha and beta chains. But in case of DR, that's an exception where only the beta chain is variable, and we mention only the beta chain here, like HLA-DRB1. So coming to class 2, compared with class 1, it's for class 2 it's a bit more challenging to develop binding prediction algorithms. One reason is that within the longer peptide, we have a binding core in case of class 2 peptide binders. So there's a 9 amino acid residue core within this. So when we develop this class 2 binding prediction algorithm, we have to first correctly identify the binding core, the 9 amino acid binding core. So for that, basically it needs proper alignment of this binding core with the binding groove of the molecule. So that's kind of a bit more challenging than compared with class 1. And then there are these peptide flanking residues, as I mentioned before. So if you take a 15 amino acid long peptide, so there's a 9 amino acid core inside here, and there are the flanking residues at both ends of this binding core. And these flanking residues also take place or interacts with the MHC molecule, and they also take part in the determining the binding affinity or strength of the binding for in case of class 2. So again, that's another challenge. So we have to take the peptide flanking residues length and the composition into consideration like when we develop the binding prediction algorithms for class 2. And a couple of other challenges that we face are like availability of the data set. So like Bjorn and Sandeep said, well, in case of class 2, the data set that are available for more broader alleles are less. So there are some frequently studied alleles like O2, O1, O1. So we get more data for those particular alleles, but for the rare alleles, we don't have much data. So we need data. We love data. So from perspective of people like us who develop algorithms, we love data and we need more and more data. So that's why we request everybody to deposit all the data in IEDB, or just let us know so that we can use the data to improve our prediction methods and develop new methods. So since data is comparatively less for the class 2 binding prediction tools, we have fewer number of alleles, fewer number of species available, and also the prediction performance is not as good as class 1. It's not bad, it's good, but it's not, compared to class 1, it's not as good as class 1. Yeah. So this is what I was mentioning, like, if you compare the class 1 and class 2 prediction methods, so when you have like 0.9 in case of class 1, class 2 is like around 0.78 or 0.8, somewhere around like that. So it's not bad, but compared to class 1, it's slightly less. And one more difference, in case of class 2, the peptide length is fixed at 15. So this is for the tool, not that the MHC class 2 molecules can bind only length 15. No, it can bind with lengths or different lengths, but for the tool that we have right now, the length is fixed at 15. But soon we will be changing that. We will be able to accept more different lengths. We will be updating it. Right now, NetMSC 2-pan tool can accept different lengths, peptide lengths. So we will be implementing that in our tool also. But for now, for the time being, it can accept only lengths of 50. So that's, again, I'm repeating, it's just for the tool, not that the molecule can bind only with 50. So like I said, since the performance is slightly less for class 2, we recommend slightly higher threshold, binding threshold for selecting the binders for class 2. So I will go in detail about the threshold and all of the details in the coming slides. So Sandeep already showed the methods available for class 1. So in case of class 2, these are same, like corresponding to, so we have the same methods corresponding for here for class 1. So the first one is consensus, which is in turn a combination of 3 different other methods, like it's SMM align, NN align, and combinatory library or Stoney. So for the alleles that we have, prediction algorithm for these methods, we will use consensus. And if not, we'll be using NetMSC 2-pan. So this is a neural network based method. And these are the references for these different methods. So it's there, so you can I'm not going in detail about all these methods, but it's there, and if you want to go in detail, you can refer that later. So, and these are the performance metrics in terms of AUC, area under the curve from ROC. So this is a rough guideline. So when you compare these AUCs, you need to be slightly cautious because the data sets may be different. So you cannot directly compare these different benchmarks. They come from different studies. So you may not be able to compare them as same, but it's a rough guideline, where they do stand. And the other methods are like based on artificial neural network. We have NN align, SMM align, which is based on stabilization matrix alignment. Then there's combinatorial library and true neural. So these are the different methods that we have available on our tool website. So now that was a brief introduction about the Class II binding prediction tool. Now let's go to the web version of the tool. And yeah.
I'm sorry, can you tell me a little bit more about the area under the curve measure?
So that's a method of evaluating the prediction methods. So we have the ROC, so for different thresholds.
So this is a distribution?
Yes.
And then we're looking under the area that's covered under this distribution?
Yes.
And distribution of what?
Beyond. Yeah.
So this was what I was showing before. This is AUC is it's typically used in the quality of diagnostic tests in clinical studies. So when you plot specificity versus sensitivity, you're getting this curve, and the area under this is the AUC value, where 0.5 is a completely random prediction, where your rate of false positive and true positive is the same, versus 1.0 would be perfect. And one interpretation of this is actually if you have two peptides randomly, and what is the probability that the one that is a binder has a better score? So 1.0 you would have perfect accuracy in your classification, 0.5 you would be completely random, you'd be flipping a coin.
Just a quick question, why net MHC2 PAM 3.1 is not in the consensus? Can you add it? It seems to be a performant method.
Yeah, so this was set based on a study, like sometime back we had comparison, and consensus was found to be performing better, and now NetMSC PAN like being retrained and is performing better. So we may be updating it, So that's why we recommend to select the recommended method. So once we find which method is performing better on the latest benchmark studies, we may be updating, changing the recommended method, and then we'll be using that one. So let's move on to the web version of the binding prediction tool. So this is same as all the other tools, like the basic framework is same format. So at the top you can find, so the link is tools.idb.org MSC2. And you can also come here from the homepage of IDB website database. And at the top you can find all these different tabs, and here you provide the sequence. You can either copy paste the sequence in FASTA or plain sequence, or you can upload your sequence as plain text file. You select the method here and the alleles that you want, the species and locus. So we have only two species available for Class II, human and mouse. And then further you select the alleles. So there are different options. We will go through that in detail in the coming slides. And then also email address at the end. So it's better you provide the email address, because if it's there's a lot more data, it may take longer and sometimes it may run out. But if you are providing the email address, you will get the results in your email. So, and it stays in your email, right? So you don't lose the prediction results. So it's always better to provide the email address. So once you provide the sequence, these are the methods that we discussed before in the previous slide, and so you can select which method you want. So normally we recommend selecting the IDB recommended method. So right now it's consensus, which is a combination of 3 methods, or NetMSC2 PAN. So we will change the recommended methods based on new benchmark and how the different methods perform. So like Sandeep said, these are the different advantages. We will use the best available methods, and it will give a consensus percentile rank, and it will also give the binding affinity and percentile rank for each of the component methods which was used. And coming here, next step is to select the species, locus, and alleles here. It's all basic things. You are already familiar with that. So one thing you can note here is that sometimes when you, by default, when you drop down here, you will see previously defined combinations of alpha and beta chains. So, but in case you have a different combination, you don't see it here, you can make it on your own. Like if you check this box, you can select alpha and beta chains separately. So once you select this alpha chain, you will get a combination for the beta chains available. So you can make that combination. So yeah. And another default option is like reference. So this is the 27 alleles that we that Sandeep. In case of this is in case of class 2, Sandeep was referring to a similar set of 27 alleles. So sometimes we get this question, like some people maybe starting with these things. So what allele should I be using? So they don't have a specific population, or they don't have an allele typed subject or donor population or anything like that. So they are confused which allele or what allele set we should be using. So this is what we recommend. So this is a set of 27 alleles which will give a kind of global population coverage. 97% of the global population will be covered. Not all the population will be covered. Like there will be some rare populations or some countries which may not be covered, but kind of gives a global population coverage. So to make this selection easier, there's an easier option, checkbox. You can check that box and this will be automatically selected, this 27 reference set of alleles.
Do you allow for DRA polymorphism?
What's that?
Do you allow for DRA polymorphism with the DRB1?
What do you mean? Sorry.
Can I put in a genotype? Can I put in the DRA and DRB1 in the same?
For the DR, it's only DRB is accepted.
Yeah.
Is that
so there is a polymorphism in the
in case of
DR
DRA also?
which
Yeah.
is not being, we don't take into account that at all.
Yeah.
Because unfortunately, all the data we have for binding assays, the alpha chain is not typed.
Okay, so I'm
essentially the
working with.
entire data we have in the database, we don't know what the alpha chains are, so we don't know the impact of the alpha chain on binding for DRB. For the others, you can. I think Simon Shang is showing that in the book. Yeah.
So I'm working with the Pima Indians. We have a really nice polymorphism, a 2-allele polymorphism in DRA, a 101 and a 102.
Yeah.
And they both have a fairly high frequency, so that
Okay.
would be, that's
Yeah. We'll be interested to see all those kind of data so we can improve on this. Yeah, thanks.
So you mentioned that there are like something not in the reference set, right? There are some like some allele is not in the reference set.
The reference set has only 27 alleles, which are the most frequent alleles in the global population.
So
if you want, like if all the other alleles that are available for prediction, you will get on the drop-down menu.
Okay.
Yeah.
if the allele's not on the drop-down menu, then it's not.
No.
So I think I looked for 1402, for instance.
Mm-hmm.
The RB1 1402, and I'm not sure that I
Are you
found.
showing the pan predictions in a second?
Yeah, and let me quickly go to that website. So. So 1402. 1402 should be here. And it's here.
Ah,
And the NetMHC pan method can accept like a wide range of alleles because it's a kind of pan method. So that's the concept, right? It can accept it.
yeah, I didn't see that the last
Yeah.
time.
And again, like we are updating this
Oh, okay.
every now and then. So. So more details are available on the help page about this reference set. And so there's one more set. It's a 7 allele set, so I will give more details about this 7 allele in later slides. So I'm not going in detail about this here. But there's a checkbox here for quick access. And again, like if you have a specific set of alleles that you want to do the prediction again and again for repeated analysis, you can save that as a plain text file and upload that allele file. So for example, you have a lot of different data sets. You don't have to go to the drop-down menu and select every time. So you can save that as a plain text file and upload that allele file. So just remember to follow this allele name format, nomenclature format. So if the format is not different, the tool may not recognize that. So just keep in mind. And it can accept only the allele names, not the allele sequences. And only available alleles for which we have prediction method. So now you know how to upload your data in the tool. Let's see how the tool works. So again, how the tool works behind, it's same as the class 1. So once the tool has the sequence, it breaks down the sequence to all possible 15 peptides. And then predicts the binding affinity for each peptide based on the method that we chose, and then compares the predicted binding affinity with a large set of random selected peptides, and then assigns a percentile rank depending on the individual predicted affinity. So it's all same as before. And in case of consensus method, it's a combination of 3 methods. So we take the median of these 3 methods. Again, we discussed about all this in the previous session, so I'm not going in detail. So yeah, percentile rank once again, like it's generated by comparing the selected peptides predicted binding affinity against that of a large set of randomly selected peptides, and it provides it does is basically provides a uniform scale, whether you want to compare between different methods or different peptides. So it provides a uniform scale from 0 to 100, and basically ranks the peptides from on a scale of 0 to 100, based on which you can select the top peptides. And a lower numerical value, lower percentile rank, indicates higher affinity. So now I'm taking you through kind of an example how I would do this binding prediction. So I have the sequence from HCV virus here. I am selecting the recommended method and selecting human, DP locus, and this allele. So I'm going to do the binding prediction for this allele for this sequence from hepatitis C virus. And this is the result that I get. So on top, it's again basically same as class 1 prediction tool. On top, you can see the input sequence. You can download the result as CSV file. You'll see the citations, and that's another reward that you can give us. Whenever you use these tools, please cite our papers. So that's the only reward that we are getting. And at the bottom, you can see the table, the prediction results. So in the first column, you see the allele. Second column is the sequence number. We have only one sequence, so that's showing 1. And then there's the start position of the peptide within the sequence, end position, and the peptide sequence, and the method that was used, and the percentile rank. Again, it's sorted based on the percentile rank. The top peptides are coming at the top. The lower value means better binders. And so since this is a consensus method that was used, there are 3 methods used, again same as class 1. So if you check this box here, you can see the different values, metrics from different component 3 different methods. It shows the predicted IC50 and predicted percentile rank. And again, consensus percentile rank means it's the median of the 3 methods. And this is the downloaded result in CSV format. You can use this to filter or screen using a spreadsheet, or you can use a script. And this is the email. Once you provide the email address, you will get an email like this, just like class 1 that we saw. So coming down here, you did the prediction. Now you have all the prediction results for all your peptides. And how do you select the binders? Again, like the previous class 1, we are recommending to use IEDB. So based on what scale do you select? So what we recommend is to use the percentile rank, IEDB percentile rank. It's a scale from 0 to 100, which essentially ranks all your peptides based on the binding affinity strength. So cutoff guidelines, in case of class 1, we recommend a lower percentile rank threshold, but here in case of class 2, we recommend higher threshold, which is 10. And MHC IC50 in case of class 1 is what we recommend is roughly 500, but here we recommend 1000. Yes.
Which IC50 value we should consider? Means SMM align or NN align? because both these have different values.
Yeah, so that's why we are recommending to use this one.
No,
So,
here you
yeah, I
have-
know this is different.
Yeah.
The IC50 values are different, so that's why you may get confused. So that's why we are recommending use the percentile rank.
Instead
So
of
and
IC50.
instead of IC50, that's what we are recommending. So the percentile rank essentially is derived from this the predicted IC50 for each method, and we combine these three methods and derive a consensus percentile rank. So unless you are really want to look at these scores and you want to specifically follow something, unless you don't want to go that way, you just follow this one, the percentile rank. So this is basically ranking peptides based on its predicted affinity strength. Okay. Yeah. So if you don't have a specific, I'll come in more detail here. So talking about the binding affinity cutoff or threshold, we generally in the previous slide, as I mentioned, we generally recommend a cutoff threshold of 10. So that doesn't mean that that's a hard and core cutoff, like a peptide with 10.01 is a non-binder and a peptide with 9.95 is a strong binder. It's not like that, right? So that's a general cutoff guideline. So depending on your needs, like different people working with different studies will have different needs. Sometimes you will need a large set of peptides that you want to study. Sometimes your resources may be limited and you want to select only small number of peptides. So what we ultimately provide is we predict the binding affinity of each of the covalent peptides in your sequence, and we rank all these peptides on a scale 0 to 100. So depending on your needs, you can select top 10 peptides or top 100 peptides based on this scale. So essentially what I'm saying is, like, you can change the threshold, higher and lower depending on your needs. And sometimes you may need to select a specific number of peptides for each of the alleles. For example, you're studying 10 alleles and you want 10 peptides each for 10 alleles. So if you select 10, use a cutoff of 10, you may not get enough number of peptides for each allele. So based on your requirement, you can adjust the threshold. But ultimately, the message that I want to convey is that this is what we are doing. Like, we predict the binding affinity for each of the peptide in your sequence, and then rank them, and then you can select the peptides based on your needs. So if you don't have a specific number or resource limit, and you're confused what cutoff I should be using, 10 is the generally recommended cutoff. So that will select almost all the top binding peptides. So, and now you know how to provide your sequences, do the prediction, and you got the binders. So if you submit a sequence as such, as a whole, and when you look at the results, prediction results, this may be what you are getting. So when you look at this result closely, you will see that these top peptides, so these are the top peptides coming from this sequence, like top percentile rank. You will see that these peptides start at like 526, 527, 528, 529. So essentially, they are redundant, right? So you don't need to synthesize all these peptides. It's redundant. So why it's like that? Because the binding core within that is same for different methods. It's same. So we don't need to do that, right? So in order to avoid that kind of a redundancy, what we recommend is, like, instead of submitting the entire sequence as such, just pre-process the sequence into 15 minutes overlapping by 10 amino acids. So this will largely avoid getting the redundant peptides in your result. So why we use 15 minutes overlapping by 10? 15 is not the only length, but it's the mostly preferred length for class 2. And keeping a 10 amino acid overlap means you will get a minimal number of peptides with a binding core and at least one residue, flanking residue on both sides of the binding core. So that's why we recommend 15 overlapping by 10. But you can also use your own criteria, maybe like 16 more overlapping by 10 or something like that. But this is what we generally recommend, and this is what we follow here mostly. So you can use a Python Perl script, or or even the spreadsheet to generate like 15 mers from your sequence. So on the other hand, like, if you did not do the pre-processing, you submitted the entire sequence, and you get the results, then you may have to do a post-processing, like remove all the redundancy. Like, you select the top peptide and then remove all the subsequent overlapping peptides, then select the top peptide and then remove, all these things. But we have a new tool, as I mentioned, like, Epitope tool, a new interface. and I will go through that in the later slides. So this is already incorporated in that Epitope, so you don't have to worry about any of these things. So I will go through that in the later slides. So so far we have been going through class 2 binding prediction for individual alleles. So now I'm just moving on to prediction of promiscuous binders. So do you have anybody has any quick questions so far?
Promiscuous binders could have been done for MHC Class I as well or not? Or is
Not-
only specific for MHC Class II?
Yeah, you do class promiscuous binders for class 1.
You can do that.
You can in
Yes.
principle do that. for
Yeah, you
class
can select
I.
the reference allele set and you will be predicting for 26 allele, and you can choose like how many peptides are binding to more than 50% of the allele that are selected.
Yeah, you can do that. Yeah.
So sometimes we can see like some protein sequence have non-standard amino acid. So would that work?
No.
Okay.
It
so-
can accept only the 20 standard amino. Okay, so promiscuous binders, what are they? They are basically peptides that can bind with more than one MHC molecule. So why we are concerned, like we give importance to them, because we have found that these are associated with stronger antigenicity and since it can bind with more number of alleles, they can cover larger populations. It can have a larger population coverage. And this is important in reducing the immunogenicity in case of therapeutic proteins. For example, you substitute one amino acid, it can have an impact on more number of alleles. And you can use these tools to predict the promiscuous binders. And here, since more number of alleles are involved, we again recommend a higher threshold of 20. So how we can do that? So essentially we are using the same prediction tool, and here instead of using one allele, we select multiple alleles. So if you have a set of alleles, for example, if you're considering a specific population or specific donor set, and you know the allele types for that specific population, you can use that one. Or if you don't have a specific population, you can use the reference set of the 27 alleles. So you get the results. It's all essentially same as before, but the only difference is you get prediction results for all the alleles included, intermingled here. And so this is the reference set of 27 alleles. That you already saw this before. So if you don't have a specific population with you that you want to study, you can use this set. And once you get the prediction results, you look at how many peptides, each of the peptide, for each of the peptide, how many alleles does this bind? So for each individual peptide, the threshold is like 20. So we are setting a higher threshold, and this is for these peptides for that particular sequence. You are seeing that this peptide can bind with 8 alleles out of the 27, and there's one peptide that binds with 21. And as a general threshold, we would pick peptides that can bind with more than 50% of alleles. So here we had like 27 alleles. So there's only one peptide that can bind with more than 50% of the alleles. Yes.
Is this a derived table from the output?
Yes.
Okay.
Yes, yeah.
Alright.
It's not
Post-processed, yeah.
Yeah, it's post-processed. So that's how we generally, predict promiscuous binders.
Sorry, when you say greater than 50% of the alleles, you mean from the 24?
Yeah, 27.
Or 27,
Yeah. Mhm.
the set of 27?
Yeah.
Okay.
So basically like more than 13 alleles. Yeah.
Okay.
And this is, now I'm moving on to another method, like this is what we call it 7 allele method. We published this paper a couple of years before. So in this study, what we were trying to find the immunogenicity. So we had data sets of immune response from different studies here. So these were the data sets that we used. And these are not binders or binding affinity, but the immune response. So essentially we are trying to find, the aim was to capture the maximum immune response with the least number of peptides, so minimum amount of peptides. And we had like 6 data sets, peptide data sets, and which contained essentially 15 or 16 more peptides with 10 amino acid residues overlap. So what we found that we tried different approaches, and what we found was that when we use a set of 7 alleles basically 3 DRB1 alleles with frequency of more than 12% and 4 DRB3 or 4, 5 alleles. When we use these alleles, and we found that around top 20% peptides could capture 50% of the immune response. And the median consensus percentile rank for the 7 alleles, when you take all the 7 alleles, and the median rank for them corresponded to roughly 20 median percentile rank. So this is a method that we recommend to select top immunogenic peptides from a set of peptides. So how you will do this? So generate 15 mers overlapping by 10 amino acids from your sequence, and do the binding prediction for the 7 selected alleles. So in the previous slide I showed a checkbox where you can select the 7 alleles quickly, and select all peptides with median consensus percentile rank less than 20, and this set of peptides can roughly capture 50% of immune response. So this again is implemented in the TEPI tool, and I will go through the TEPI tool in the later slides. And it's also implemented in a new tool developed by Sandeep called CD4 Episcore tool. So Sandeep will go over that tool in the later slide, in the later session. So right now we have some time for exercise. I think it's good if you can go through this exercise. So it's one of the simplest, most easiest questions that you can get. We have a peptide. So predict the alleles from the given set of 6 alleles that can bind with this peptide. It's easy. So maybe you can copy paste the sequence from your slide. I think share the slides with you, or it's an easy one, 15 more peptides. Just let me know if anybody has any question. If you get stuck, we have 5 minutes for trying that out. Okay, are you getting it? Okay, so we'll just go through it. So you must have copy pasted the sequence here. You must have selected the method as IEDB recommended and selected all the 6 alleles from the drop down menu. And this you should have got this one, right? So there are two alleles basically with percentile rank less than 10. So I would say these are the predicted 2 alleles predicted to bind to this peptide. Okay, so if anybody has any question, maybe we can go after this session or in the individual session in the afternoon. So now I'm moving on to the TEPI tool. So TEPI tool is basically a new interface for the prediction of class 1 and class 2 epitopes. So essentially we have the same methods implemented in a more user-friendly way. So what it does is, the motivation is like, make the tools more user-friendly, provide recommendations as a default. So sometimes users get confused what threshold I should be using, how to select the binder. So we provide the recommended values as a default. And at the end, you will get the selected top peptides based on your input parameters. And you will also get the complete prediction, but ultimately you will be getting a concise set of results so that you don't get confused with all the prediction results. So this is in the form of a step-by-step wizard with 6 steps, provides recommendations as default values, and input parameters. Although there's the recommended default values, you can adjust that, increase or decrease. And we have also incorporated new methods here, the ones that we discussed, one Sandeep mentioned in the previous session, and also the 7 allele methods. So I'll do a quick tour. You can try it yourself, and we will be making this the default, the main tool in future. So this is still under the labs. So this has been there for 1 or 2 years now. We have been getting feedback on this tool. So once we fix everything, so it's almost ready, it's running. People are using this and it's running without any issues. In near future, we'll be moving this to the main tool section. So we request you to use this method and let us know if you get stuck with something, any issues or any bug or anything like that. So that's important for us to get feedback. So in the first step, you provide the sequences. You can either copy paste or choose sequence file in plain text format. In the next step, so this is for class 1. Here in the second step, you can select the species and the allele class. So for class 1, you get all these species. Class 2, you get only 2 species. And in the third step, you select the allele. So you have different options for selecting. So the first one is by default, select from list of most frequently occurring alleles. By default, it's alleles that occur more than 1% in the global population. And the second one is select from list of available all the alleles. So if you don't see your allele that you're looking for in this one, you can select the next option, so that will list all the alleles that are available. And so there are HLA allele super types. So if you want to select alleles from this selector, representative alleles from this HLA super types, you can choose this option. And this is the panel of 27 most frequent allele set. Or you can upload your own allele file. Again, remember to have allele names in the proper format. So all your selections so far will be displayed on the right side. So you have 3 sequences. Species is human, the class is actually class 1, and these are the selected alleles so far. And in the next step, for fourth step, here you select how many peptides you want. So by default, it's default settings for low number of peptides. So low number means we are removing all the duplicate peptides, and the length selected is only 9 more. So, and this shows an approximate number of peptides that will be there. Like when you select 9 mer from this peptide, you will have 58 9 mers. And then there's also one option where if you want to do conservancy analysis. For example, if you have a set of homologous sequences and you want to see how conserved this peptide is in the set of sequences, you can choose this option. In the second option, is apply default settings for moderate number of peptides. That means all the lengths from 8 to 11 will be selected, 8, 9, 10, and 11. And the third one will be default settings for high number of peptides. So we have 8 to 14 mer, that's the available lengths in class 1 prediction tool. So all those lengths will be selected. And this is the custom selection. This option is for custom selection. where you can select your own length. If you want to select 8 and 9 or 10, you can go this way. And in the fifth step, here's where you can select the method. So at the top, you can see, choose the method, or recommended method, or NetMSC pen, or any other method. And here, how you want to select the predicted peptide. So if you want to select peptide based on predicted percentile rank, that's what we recommend, and that's the default option. You can select that one. And if you want to go with the IC50, you can select the second option, and the recommended value is 500 nanomolar. It's given here, but if you want to adjust that, you can do that here. And this is the allele-specific binding affinity, which Sandeep was showing in the previous session. So these are the alleles for which we have this allele-specific affinity. So if you want to predict for any of these alleles, and you want to choose the alleles MHC-specific binding threshold, allele-specific binding threshold, you can use this option. And if you want to select a specific percentage of peptides from your data, for example, if you want to select 2% of peptides for each of the alleles that you used, you can go with this option. So normally we will have to do, like, after you get all the results, you will have to calculate, okay, how many peptides are, like 4 or 5% of peptides for each allele, then you will have to select it. So you don't have to worry about all those calculations and selections. So if you choose this option, by default it will select the needed number of peptides, percentage of peptides for each allele and each sequence set. And this is based on the number. So the previous one was percentage based, and the next one is specific number. So if you want to have 5 peptides per allele, you just mention how many peptides you want here, and then you'll get 5 peptides per allele in the final results. So in the last step, 6th step, you can review your selections and submission, and at the end, you can provide a job name and email address. And I think this is important to give your email address so that you get your email results in your email. Plus, if you provide a job name, we can, if you stuck with something, if there's an issue, you don't get the result, we can look up in the log and then determine what went wrong and all those issues. So we recommend, it's optional, but still we recommend providing an email address and a job name. And so once you submit your input sequence and all the parameters, this is what you get on the web page. The results displayed on the web page is like this. So at the top, this is the predicted results based on your selected input parameters. So there are 3 peptides selected. So we provided a percentile rank cutoff of 1. So there are 3 peptides with percentile rank cutoff less than 1. And then the conservancy. So this is the conservancy of this peptide in the 3 sequences. So 2 out of the 3 sequences we submitted, 2 had these peptides conserved. And you can download this as CSV file from this link, and all the complete prediction results for all the peptides in your sequences is available here. So you can download it here. And the conservancy analysis, you can download it from this link. So I will just show you here. And these are the input sequence and input parameters which are displayed on the result page as well. And this is what you get. So this is basically same as the default main legacy MHC Class I binding prediction result. So you have the result for all the peptides, all the component peptides, which you can download. And this is the peptide conservancy. So for each peptide, you can see which. So we had 3 sequences. It's showing which all sequences had this peptide present, and this is showing the conservancy percentage of this peptide within the sequence set. So in case you want to select only the peptides that are conserved in almost all the sequence or 50% sequence, you can use this table. So if you provide your email address, you will get something like this. So you have the selected peptides as a table here, and you can download all these. There are links for downloading these results. So the results, the tables will be stored for 3 days in our servers. So you can go to this link and download within 3 days. Yes.
Is there a web API?
No, not for this. I mean, basically we are using the API for, so we have API
Behind
for
the
prediction.
scenes.
So we are basically essentially using the same APIs for.
Oh, okay.
Okay. So so far we were going to the class 1, and there are some differences for class 2 in the steps in between. So I will just go through those steps where class 2 is different from class 1. So again, so this is step 3 where you select alleles. So there are 3 options. Predict for custom allele set. So there's one option here. You can select from the list of alleles. This is essentially same as before. Like you're familiar with that right now, and or you can upload allele file here. And there's another option here, predict for pre-selected panel of alleles. So here we have a panel of 20. So here, when we were making this tool, like TEPI tool, we had only 26 class 2 alleles in this reference set, but now we have 27. We will be updating it soon. So you can select. So for example, if you want to select the most frequent DR alleles, you can just use this checkbox here. And most frequent BP alleles, you can use this. So it's just to make easy, selecting alleles easy. So nothing more than that. And there's one more option, predict using pre-selected allele sets and methods. So this is the 7 allele method where you can select. If you go by this option, by default, this will use the 7 allele method. So ultimately, you will have predicted peptides using the 7 allele method. And again, coming here, this is the panel of 26 most frequent alleles. Yeah, and the next step is where you select the number of peptides. So here, the peptide length is fixed at 15. So you don't get option like class 1 to select different lengths, but the length is fixed at 15. But what you can adjust is the number of overlapping peptides, overlapping amino acid residues. So by default, this is the default selection, apply default settings for moderate number of peptides, and it's fixed at 15 mers, 15 amino acid length, and peptides that are overlapping by 10 amino acid residues. And if you go by this option, there will be approximately 12 peptides generated from your sequence. And the other options are same, like conservancy analysis. If you want to do conservancy analysis, you can select that one. So if you go by apply default settings for low number of peptides, so what is different is this one here. The number of overlapping residues is reduced to 8. And if you go here, custom selection, you can adjust this parameter here. Again, the length cannot be adjusted. For now, it's fixed at 15, and you can adjust the number of overlapping residues here. So coming to this step here, in case of 7 allele method and panel of 26 most frequent alleles. So this step here, you cannot make adjustments. So this is pre-chosen or pre-selected method, 7 allele method and 26 most frequent alleles. So this is for basically selecting the promiscuous binders. So this is fixed here. Number of overlapping residues is 10, and the number of peptides that will be included is like 12. That's fixed. Again, at the 5th step, you can decide on what basis you want to select the peptides, either predicted percentile rank or IC50 or number of alleles or different parameters. So this is for the promiscuous selection. So you have the predicted results here, and if you want to select peptides that bind to at least 50% of alleles, so that's the default. So you must be remembering when we discussed predicting promiscuous binders, I recommended having 50% of alleles. So that's the default recommendation that we are giving here. And the recommended consensus percentile here was set as 20 for predicting promiscuous binders. So in case of 7 allele method here, the recommended threshold is 20. Again, you can adjust that. You have the option. And you already saw this, like the 26 most frequent alleles. So again, in case of class 2, again, you get the result like this. So these are the selected predicted binders, top binders, and here you can see one extra field here, that's non-redundant results. So when you go with complete results, you get the prediction results for all the component 15 mers. But non-redundant means the peptides that are overlapping by 10 amino acids. So it's kind of we already post-processed and removed the redundant peptides. And the conservancy, you already saw that. So this is the results in the CSV format and post-processing that I already mentioned. So we already removed this redundancy. So you don't have to worry about that. So that's about the TEPI tool. So I think it's kind of intuitive, and you can go and try it out and let us know if you have any question or you get stuck with something, and we'll be happy to answer. So right now I'm again moving forward. I'm going to explain 2 more other versions that we have. So far we have been going on the web version, and then we have two more other versions, so which are a little bit more kind of a computational for computational persons. So first is the API, or Application Programming Interface, or RESTful interface. So what it is basically it's a programming interface where the client can submit the parameters on your machine, but these parameters are sent to the server here. So the prediction is basically done here, although you are using command line interface, and we do the prediction at our servers here and send the prediction results back to your computer. So it's kind of a different interface. So we have this API service for both Class 1 and Class 2 tools, and also some other tools, analysis tools. And as I mentioned, it sends the request or parameters to the servers at LGI, and the prediction is done based on your supplied parameters and sends the results back to your computer. So why we should be using this? What are the advantages? So no need to install tools on your machine. So there's another version called standalone, which you will have to install it on your machine, local machine. And this is you don't have to, when you use this one, you don't have to use the web interface. So if you have to do a lot of repetitive analysis, repetitive predictions, you don't have to go to web interface and then select your alleles and all fill the forms. So you don't have to do that. You can customize this queries or the commands, and you can easily do the repetitive kind of analysis. And this is freely available to all users. I'm saying this because we have licensed version of for industry. There's a version of like the virtual machine which provides all these tools as a virtual machine, but this one is freely accessible to everybody, no license or anything like that. And this is automatically updated, so we update it here roughly once in 6 months. So you don't have to worry about updating. So if you have the standalone methods, standalone tools, or you have the license, you may have to update it as we update the tools. But for this one, we update it at our end, so you don't have to worry about it. And the main advantage is like you can build your own scripts, your own tools using these APIs. So you have your own sequence set, you have your own pipeline set up, so you can plug these commands or parameters inside your script, and you can run the script or pipeline. And you can provide multiple sequences and alleles or peptide lengths like with different combinations in the command. And this is much easier in case of when you have larger data sets. So sometimes when you supply too many number of sequences or select too many alleles, the web version can crash sometimes. It may not be able to handle too much data. So, but in those cases, it's better you go with API or use the standalone method. So more details are given here. So this is the main homepage of the analysis resource. So at the top you will be seeing like all these tools, like overview, T-cell tools, B-cell tools, analysis tools. So one of the tabs is Tools API. So all the details, what all methods are available, and sample commands are given here. So you can try them out. And if you have any question, just email us and help@idb.org. So sample commands are given for Class 1. So this is for run a single allele, single length combination. If you want to run multiple allele and length combinations, you can. There are different sample commands here given here. So you will have to use a terminal, to run these commands, Mac or Linux terminals, and then you can try them out.
Do the results live on your machines for a little bit, or are they like sent back and then?
What's that?
Like, are any results stored locally on your machine when you use the API?
You can store it. I mean.
But on your machine.
No, I don't have. This is not my computer.
No, but when you use the API.
They're stored for about 24 hours.
Oh, okay.
24 hours.
Yeah.
Okay.
Sorry.
So I have a question. So you mentioned that for the people who license the IEDB database, for the people who license IEDB database, they have to update their tool.
Not the database, the tools. So
it's the tools. Okay.
Yeah.
So does so for the people who license IEDB tools or even I don't know if it's also database, but do they have API available in at their end, or?
If they license the virtual machine image, then yes.
Okay, so they can basically use API to access their own server to around there. Okay, thank you.
Is this all Linux based?
Yeah. So this is a sample command for class 1. So basically you have the method here, sequence text here, allele and length, and an email address if you want, and then this is the server address. So you'll get essentially the prediction results like this. And this is for class 2. The only difference is we are not providing the length here, but essentially all the other things are same. So that's about the API version, and I'll quickly go through the standalone version as well. So again, you can download this the standalone method from the download page. So for each tool, there's a download tab, and if you go there, you can find the download buttons and all the details provided here. So this is available for class 1, class 2, and some other tools. And on some Linux, we discussed about the licensing, and I think there's a PDF provided along with the workshop material, much more details about the licensing. So if you have any question about licensing, please go through that, and or contact us at J. And there's an email address, right? license@idb.org? Yeah, license@idb.org. So if you have any question, please email. So we'll be answering any question.
But also to stress, like academic use is completely free.
Yeah,
right?
for
This is
academic
specific.
non-profit use, it's completely free. And you can, again, we will provide support. You can contact us, but for the licensed version, we'll have like dedicated support and all these updates and everything. So we'll be, when there's update, we'll be contacting you. Like we'll be sending out emails to let you know that okay, there's updates coming up. So why you should be using standalone? When there's large data sets, it's better you use standalone. You don't have to go to that web version, and you don't have to depend on the internet. You can implement the tools on your own machine when there's large set amount of data, when there's repetitive analysis you have to do, and free for non-profit and academia, and available for industry at a license-free fee. And especially in case of industry, when you have proprietary sequences and all, you may have to keep it secret, so you can get the license and use it in your own organization. So this is a sample command for class 1. You select the method. This is the script or the tool, and then select the method, allele, length, and your input file, and you'll get the results like this. You can store this as a output file by providing an output file name. And this is for class 2. Everything is same except we are not providing the peptide length. And all the detailed instructions are available in the README file, which is downloaded along with the tools. And if you have further questions, please contact us. So we have a policy of quickly responding back to you when you send out a question. So we'll be contacting you back in 48 business hours. And if there's more detail we need, we'll get back to you, or we'll discuss among us, and then we'll get back to you. So please contact us if you have any question. So this is a summary of different versions. I'm not going through this in detail, but you have the slides, so if you have any question, you can just refer this or get back to us. So in summary, we went through class 1 and class 2 tools. We did have a brief comparison. We learned how the tool works. I gave you different recommendations, methods, which method to use, what kind of thresholds, and how to do the prediction of promiscuous binders. And we saw the 7 allele method, and we quickly went over the TEPI tool, and we also took a look at the other versions, API and standalone. Thank you. So we have time for maybe a couple of minutes for some quick questions, or we have individual sessions in the afternoon like we had yesterday, so we can take your specific individual questions, or if you want to do any specific predictions or anything, we can try that. Or if you have any quick question, I can take a couple of them.
So tell me about how does the TEPI tool work in comparison to just the standard interface? Is it just more convenient?
It's mainly it's the convenience thing. So and essentially for some new people who want to try, so sometimes we get a lot of questions. What threshold we should be using, what alleles we should be. So basically, we are providing some default recommended values so that they can try that. And essentially, the prediction methods same, no difference. And plus we have incorporated some new features like new methods like 7 allele method and allele specific affinity, all those kind of things. And then post processing, or you don't have to worry about the redundancy on all those things. So a lot of that convenience features. So yeah.
Yeah. Why did you choose DRB 3, 4, and 5 for your
7
some of
allele.
your standard sets? For instance, 4 and 5 are not present on
Yeah,
the
so we
HLA
did a
loci,
study. So more
haplotypes.
Yeah, so more we did a study, and that more details are in that paper. So if you are more interested, you can go to it. But in brief, we tried different approaches. We first started with more allele sets and the 26 allele reference set, and then we narrowed down. We excluded some locus and tried. We were essentially trying to see which set and can pick up the top immunodominant peptides that can give the maximum amount of immune response with least number of peptides. So this is the set of alleles that we gave the best performance. And this is not like super perfect or anything like that, but this is what we get the best. All right, I think
10-minute break.
Yeah.
So get back at 11:15, so 9-minute break, and we'll try to be quick because as we have seen, it's very